Hello learners, today we are going to discuss about pharmacognosy phytochemistry second in their unit fifth, that is basics of phytochemistry. What are the basics of phytochemistry? So first we have to know the definition for phytochemistry. So phytochemicals means what? So this phytochemical is the Greek word. Phyto means plant, and chemistry means the chemical compound present in the plant. So these are the definition of this phytochemistries. Phytochemicals are naturally occurring and biologically active chemical compound which is present in plant is known as a phytochemicals. Then these phytochemicals generally present in crude drugs. So next is the isolation method for the crude drugs. So various extraction method, isolation method, separation technique is there for the separation of this component. So what is meant by extraction? So definition of this extraction, it is the process of isolation of soluble material from insoluble residue which may be liquid, solid by the treatment with solvent on the basis of this physical nature of the crude to be extracted. So those are the extracted material. It may be solid, liquid or semi-solid in nature. So this extraction process is totally depending on the choice of solvent, choice of crude drug and the method of extraction. So there are several modern methods and conventional method and modern method. So before that, for the extraction purpose, means for the extraction, there is the basic principle is what? Mass transfer. So mass transfer means what? Mass transfer from solute to solvent, means solid to liquid, is known as a mass transfer. So what happened in this case or in mass transfer? Firstly, drug particles immerse into the solvent. Then these solvents are penetrate inside the cell wall. Then diffusion is there. Diffusion is occur. So this diffusion dissolve the solid material into the liquid. So it solid converted into the liquid. This process is known as a mass transfer. So what are the properties for the selection of solvent in case of extraction? So those are the solvent used for the extraction. In Greek language, it is known as a menstruum. So they must be have a low toxicity. They are inert in nature, non-inflammable. It should have a preservative action. Means if we use the water, then this water does not have a preservative action. But if we add the alcohol, somewhat alcohol into the water, then here there is a it is converted into hydroalcoholic form. So this hydroalcoholic solvent is used for the extraction purpose. Then promotion of rapid physiological absorption of extract. Then means those solvent we use or those menstruum we use, they have a rapid absorption and penetration action. म्हणजे suppose drug घेतला crude drug, तो सॉल्व्हेंट मध्ये टाकला, त्याचं पेनिट्रेशन किती होईल म्हणजे त्याचं ऍब्झॉर्प्शन किती होईल यावर पण सिलेक्शन असतं सॉल्व्हेंट्स. देन ease of evaporation at low heat. So at room temperature it should be evaporated fast. So these are the some properties for the selection of good solvent. Then general extraction of herbal plants. So plants means raw material which is obtained from the plants. Before that, we have to authenticate the selected plant. So where from we authenticate the this plant? So we authenticate this plant by the through the by the taxonomist or the botanist. They compare your means they compare the our plant with available literature, and they gave a specimen number to us. Then this raw material is collected from the same species, then dry it completely, then pulverize the root drug. So here is the size reduction. If you have a bark, then by using pulverizer or mixer, grind it, then it is in the form of reduced size. So this process is known as size reduction. And this size reduced drug or the fine particles or the core particles of the drug that is further used for the extraction. So this is about the general extraction of, means this is the guideline how to collect the drug, how to reduce the size, and which drug should be selected for our extraction.